How's it going, everyone? This is genetic material replication and function. This is the first topic of the semester. I'm still kind of learning this whole format, but I hope to be able to share some of the information that I've learned, and hopefully you can learn as well. My name's Herman, and this is for my genetics course. So without further ado, let's jump right into it. So we start with the discovery of DNA. It was actually discovered in the 1800s, quite early, contrary to, or I guess what is common knowledge. Uh, its importance wasn't really discovered until well into the 20th century. Um, but obviously, DNA is the building block of life. Without DNA, um, we wouldn't be here today. We wouldn't know where we came from. We wouldn't understand our ancestry. We wouldn't understand genetics in and of itself. DNA's importance, um, it is the foundation of genetics. It's the foundation of inheritance, passing on genes, uh, genes themselves, understanding genes, understanding the processes that take place in our genes, and among many other topics, it could all be led back to DNA. And mutability is a topic I'll be discussing at the end of this presentation. But DNA's mutability is possibly its greatest asset. DNA of itself is sort of a set of instructions that it has left for itself. Um, or whoever created DNA, right? They left a set of instructions for a cell to function, for a cell to perform certain tasks. It holds genetic information for the cell, um, genes to pass down to offspring, and many other things that it holds. But it's basically a set of instructions, and its immutability is possibly the greatest thing that could have happened to humanity. I think mutability, its ability to change, its ability to be altered, uh, gives it an edge, I guess we could say, in evolution. Uh, its ability to adapt to environments over time. Uh, Case in point, the Galapagos Island, right? How birds have adapted to certain areas of the island with certain types of beaks. It's the same principle. DNA allows that to happen. So I believe mutability deserves its own entire slide. So we'll be discussing that further, but it is a beautiful notion in my opinion. And that brings us to our next topic, which is Griffith. Frederick Griffith, brilliant guy. In 1928, he conducted an experiment to investigate the transformation of non-virulent bacteria into virulent ones. Um, sorry about that. Uh, Griffith was working with Streptococcus pneumoniae bacteria. So basically, it was like tuberculosis or something like that. It was a very nasty thing at the time. People would die from this all the time once they got it. So it was either virulent, smooth, or a non-virulent strain, which was rough. He was basically interested in understanding how non-virulent bacteria could become virulent. So he designed his experiment to test that hypothesis, and he worked with mice. He was looking for the transforming principle. So he exposed a substance produced by the virulent bacteria to the non-virulent bacteria. Griffith prepared cultures of non-virulent, rough, and virulent, smooth bacteria. He heat killed virulent bacteria to create a non-infectious preparation or solution. And then he mixed the heat killed virulent bacteria with non-virulent bacteria. And he injected these mixtures to the mice that he was working with. And his results were as follows. Mice injected with a non-virulent bacteria alone did not die. Mice injected with heat-killed virulent bacteria alone did not die. Mice injected with a mixture of heat-killed virulent uh, bacteria and non-virulent bacteria died. So, in conclusion, non-virulent bacteria had been transformed into virulent ones. Um, the non-virulent bacteria had been transformed into a virulent strain by exposure to the heat-killed virulent bacteria. A substance was produced by the virulent bacteria, and that was responsible for the transformation. Um, and this experiment basically showed that DNA was the transforming principle, though Griffith didn't really discover that quite yet. Um, its significant impact on molecular biology um, was, without a doubt, um, not not merited. Without a doubt, it wasn't received without merit. It had transformation is possibly one of the founding principles, we could say, of um, kind of like DNA for genetics. So his experiment established the concept of transformation, which is the process by which non-virulent bacteria can become virulent. And it also paved the way for the next set of scientists that we're going to talk about, Avery, McLeod, and McCarthy's experiment. So their experiment was the one that actually identified DNA as the genetic uh, precursor, I guess we could say, the genetic uh, father. So, in conclusion, well, I guess we'll move on to this next set of folks. That brings us to Avery, McLeod, and McCartney, and these guys became the fathers of some of the greatest music that was ever written, with songs like "Hey Jude," "Here, There, Everywhere," uh, "Lucy in the Sky with Diamonds." No, I'm just kidding. No, Oswald Avery, Colin McLeod, and Macklin McCarthy conducted an experiment to determine the chemical nature of the transforming principle, a substance that could transform non-virulent bacteria into virulent ones. Um, and so they continued the work that Frederick Griffith had already established. Their experiment was a little bit different. They worked with sort of like blenders to work with this Streptococcus pneumoniae bacteria. Well, pneumonia, that's what the disease was, not tuberculosis. Or maybe it led to tuberculosis later on. So they isolated the transforming principle from virulent bacteria by extracting the bacteria with various chemicals. Um, they purified the transforming principle using various methods, including centrifugation. I don't know why I'm struggling with that word so much. And precipitation. So Avery McLeod McCarthy characterized the transforming principle by analyzing its chemical composition and properties, um, and this resulted in them discovering that the transforming principle is in fact DNA. DNA is responsible for transformation. So the Avery McLeod and McCarthy experiment was a landmark study that demonstrated the role of DNA in genetic transformation. Their findings provided strong evidence for the idea that DNA is the genetic material and paved way for further research into the structure and function of DNA. It's important to know, I guess, in life as well. This applies that if you work together in groups, if people come together and continue the work that others have already started, you could either come to a conclusion or further better the work that has already been done. What I mean by conclusion, it could mean anything that ranges from uh, finishing what the intended purpose of the experiment was, or finding an alternate purpose. Or maybe it's working with diseases and you finally find a cure. Or maybe you find an alternative form of medicine, right? So just a different way to treat without as many side effects, etc., etc. And this applies to, I believe, all facets of life. It took time for us to get uh, buildings right? The engineering behind it. But just I think it's something worth mentioning. Um, and so now we have the Hershey Chase experiment, and unfortunately, no, the this wasn't uh, a chocolate bar, right? It wasn't the cookies and cream that we love so much today. So they were working with whether DNA or protein was the genetic material responsible for transmission of traits from one generation to the next. Um, yeah, so back in the day, these people believed, or these people, scientists believed that proteins were the building blocks of life, that they held the key to everything. They controlled enzymes, they built the pyramids, basically everything you could be traced back to proteins. Obviously, we now know that's not the case. We know that DNA is the precursor to all things, and it gives rise to proteins themselves. Um, what they did is they worked with the T2 bacteriophage, a virus that infects bacteria, to study the transmission of genetic material, and they labeled the phages with radioactive isotopes. Um, so radioactive phosphorus 32P labeled the DNA of the phage. Radioactive sulfur 35S labeled the protein of the phage. So Hershey and Chase infected the bacteria with the labeled uh, phages. They blended the infected bacteria and centrifuged the mixture to separate the phage particles from the bacterial cells. And so they then measured the radioactivity of the phage particles and the bacterial cells. And the results were as followed. Radioactive phosphorus was found in the bacterial cells. The radioactive phosphorus, which labeled the DNA, was found inside the bacterial cells. Radioactive sulfur was found in the phage particles, so it was blended off. It flew off into the outside of the bacteria, right? It didn't go inside the bacteria, and they concluded that yeah, DNA was the genetic material. It's what viruses inject. We know that today viruses inject DNA or RNA material into the cell. They don't want to inject you with proteins. Uh, I always come back, or I always talk about how COVID works, how it had this like little protein shell on the virus, right? I guess these are its legs. Had this little protein shell that was able to latch onto to your healthy cells, right? Latched onto your healthy cells and was able to tell your cells, hey, we're not we're not like total strangers. We're from another planet, but if you let us in, we'll show you how to party. And so your soul's like, well, I guess these guys seem kind of familiar. So opens up the cell membrane, lets them in, and chaos proceeds, right? We have millions of people getting sick. So protein is just the key in order to send the DNA inside. um, but yeah, it was crazy times how they believed proteins were just the building blocks of life. And protein is in fact not the genetic material. So their significance and the impact on molecular biology was also incredibly, incredibly high. I mean, these guys they confirmed DNA as the genetic material. Um, they provided strong evidence for the central dogma, um, and that states that genetic information flows from DNA to RNA to protein. Um, so now that brings us to Rosalind Franklin. Rosalind Franklin was a British biophysicist and X-ray crystallographer, very important, who made significant contributions to the understanding of the structure of DNA. So she was born from a family of intellectuals, and they encouraged her to pursue an interest in science from an early age. She studied chemistry at the Cambridge University. Brilliant school. She later moved to Paris. She worked at the Laboratoire Central des Services Chimiques de l'État. Then she worked at King's College, where she, of course, met Francis Crick and James Watson. Um, her research in X-ray crystallography, I think it's one of the most significant things that a woman has ever done in history. Um, aside from the discovery of the tortilla. No, that was a joke. But or was it? Tacos are great. I mean, we can't deny that. Tacos are an amazing food source. But no, so her work in X-ray crystallography eventually led to the discovery of the structure of DNA. You know, photograph 51, it has to be mentioned, provided crucial evidence for the double helix model that James Watson and Francis Crick proposed. So her contributions, they can't be overshadowed one bit. She was, of course, not recognized at the time. I believe women were still having trouble even voting at that time. But women's rights were not anywhere near what they are today. Franklin's analyzed her data and concluded that DNA was a helical structure. Um, Franklin's data and insights provided crucial evidence for the double helix model of DNA. And her relationships with her colleagues were not very great. I imagine she was a woman who was quite talented, quite gifted. Um, and when a woman is super intelligent and you have a guy who's super intelligent, things tend to clash most of the time, unless there's some sort of a mutual agreement that's made, uh, like a marriage. So Franklin worked with Wilkins at King's College, but the relationship was strained. Wilkins shared Franklin's data with Watson and Crick without her permission. Watson and Crick used Franklin's data, including photograph 51, to develop their double helix model of DNA. However, they did not adequately acknowledge Franklin's contributions in their Nobel Prize winning paper. So obviously she wasn't credited. Watson and Crick sort of betrayed her. Wilkins sort of betrayed her too. I guess he must have had a thing for her, right? Something. I mean, I saw her picture. She was a beautiful woman, in my opinion. Um, so she did not receive the Nobel Prize in Physiology or Medicine in 1962, which was awarded to Watson, Crick, and Wilkins. Surprisingly, she did die in 1958 and was not awarded posthumously. So there was a severe under-recognition of her contributions to the discovery of DNA. But recently, people have come to agree that she was quite significant. In fact, they have reevaluated the situation. They now celebrate her story, and it serves as an important reminder that recognizing and valuing all the contributions of all scientists, especially women, who have made significant impacts on understanding this world. So, I mean, I think it's too late. I think it would have been great if they had recognized her when she was alive. It's too late now, but lesson learned. Hopefully, uh, if you're watching this and you're a woman, you ever make an incredible discovery, you're properly rewarded. You're given the Nobel Prize and the $1 million that come with it. Let's move on to the next subject. Moving on. Um, so we're going to talk about the structure of DNA. So this is thanks to Francis Crick and Watson and Franklin. We're not going to mention Wilkins, all right? He doesn't count. No. I guess Wilkins had his part too, that cheeky devil. So DNA is actually a double-stranded helix with two complementary strands of nucleotides twisted together, so it's kind of like this ladder rung. Imagine like little rungs in between my finger there. Uh, and we all know what DNA looks like, I'm sure. If you don't know, Google it right now. You can pause the video. and I'll wait for you if you pause the video. It has a sugar phosphate backbone. So it's composed of an alternating sugar, deoxyribose, and then phosphate molecules. It also has nucleotide bases, which in DNA are adenine, pairs with thymine, guanine, which pairs with cytosine. Um, it has hydrogen bonds between the base pairs that hold the strands of the double helix together. Um, so a nucleotide is composed of a sugar. Um, the backbone of the nucleotide phosphate group links the sugar to the next nucleotide, nitrogenous base, one of the four bases, A, G, C, or T. There are different types of bonds that hold the entire molecule together. You have covalent bonds between the sugar and the phosphate molecules, hydrogen bond between the base pairs. DNA has several physical properties. It has a double helix diameter, approximately 2 nanometers. Pitch, the distance between one complete turn of the double helix, approximately 3.4 nanometers. The base pairing, approximately 0.34 nanometers. Why is DNA structured this way? So the double helix structure of DNA solves two fundamental problems, and that one we've already mentioned before, but is the double helix provides stability to the molecule, protecting it from degradation. And its mutability, the double helix allows for the transmission of genetic information from one generation to the next while also permitting mutations to occur. So a very important detail. DNA, the DNA double helix has two grooves, a major groove, wider and deeper. Sorry about that, my computer just like completely whacked out. So yeah, DNA has a major groove, wider and deeper, approximately 22 angstroms wide. I believe that's the unit that they're using. A narrow and shallower minor groove, approximately 12 angstroms wide. So there's base pairs that are stacked on top of each other with a sugar phosphate backbone on the outside. Um, so the structure of DNA is intimately linked to its function. Genetic information storage. DNA stores genetic information in the sequence of its nucleotides. Replication. DNA replicates by unwinding the double helix and synthesizing new complementary strands. Transcription. DNA is transcribed into RNA by unwinding the double helix and synthesizing a complementary RNA strand. Translation. RNA is translated into protein by reading the sequence of nucleotides in the RNA molecule. And so, of course, this brings us to DNA replication. DNA replication is the process by which a cell makes an exact copy of its DNA before cell division. So let's briefly go over this. The process begins with the unwinding of the double helix, right? I guess we could say there's two strands now, there's four. Sorry. At a specific region called the origin of replication. So this happens at a specific place. It's the initiation. An enzyme called the helicase unwinds the double helix, creating a replication fork. There's some great videos online if you want to look or see what this actually visualize what this looks like. I recommend looking up the enzyme helicase unwinding the double helix and then creating the replication fork. And then an enzyme called primase adds RNA primers to the template strands at specific regions called primer binding sites. And then, of course, we have elongation because there's going to be adding, deletion, among other things. An enzyme called DNA polymerase reads the template strands and matches the incoming nucleotides to the base pairing rules, which is adenine to thymine and guanine to cytosine. So DNA polymerase proofreads the newly synthesized DNA strands, correcting any errors, again, deletion or any other things that need to happen. An enzyme called DNA ligase seals the gaps between the Okazaki fragments. So we have the helicase, the primase, DNA polymerase, DNA ligase. So the types of DNA replication, they're semi-conservative, The correct model where one parental strand is conserved and a new complementary strand is synthesized. Conservative replication, a hypothetical model where the parental strands remain intact and a new copy is synthesized. There is dispersive replication, a hypothetical model where the parental strands are fragmented and reassembled during replications. Okazaki fragments are short segments of DNA synthesized on the lagging strand. RNA primers. Each Okazaki fragment is initiated with an RNA primer. The replication fork of the leading strand. The strand synthesized continuously in a 5' to 3' direction. The lagging strand. The strand synthesized discontinuously in short segments, Okazaki fragments. We have the Michelson-Stahl experiment demonstrating semi-conservative replication. Michelson and Stahl's experiments show that DNA replication is in fact semi-conservative. The importance of DNA replication, genetic continuity. DNA replication ensures the transmission of genetic information from one generation to the next. And cell division, which was the sole purpose of this, right? DNA replication is essential for cell division as it provides the genetic material for the new cells in order for the new cells to function properly. So my computer glitched again. So Okazaki fragments are short, discontinuous segments of DNA synthesized on the lagging strand during DNA replication, right? They were discovered by Reiji Okazaki and his colleagues in the 1960s. Okazaki fragments have several key characteristics. They're short length, typically around 1,000 to 2,000 nucleotides in length. Okazaki fragments are synthesized discontinuously with each fragment being initiated by an RNA primer. Okazaki fragments are synthesized on the lagging strand, which is the strand that is synthesized in the opposite direction to the replication fork. Okazaki fragments play a crucial role in DNA replication. Okazaki fragments enable the synthesis of the lagging strand, which is necessary for the completion of DNA replication. The fragments allow for the replication of long DNA molecules by breaking them into smaller, manageable segments. I'll be right back, 1 second. Okazaki fragments are processed in several steps. So we have an RNA primer removal. The RNA primer is removed from the Okazaki fragment. DNA synthesis is completed to fill in the gap between the Okazaki fragment and the previous fragment. Ligation. The fragment is joined to the previous fragment by a DNA ligase. The fragments are essential for DNA replication. Okazaki fragments enable the synthesis of the lagging strand, which is necessary for the completion of DNA replication. And the fragments help maintain genome stability by ensuring that the genome is replicated accurately and completely. So now we can move on to Michelson and Stahl, which was, remember that semi-conservative discovery they made about DNA. So Matthew Michelson and Franklin Stahl conducted an experiment to determine the mechanism of DNA replication. They used a technique called density gradient centrifugation to separate DNA molecules based on their density. The experiment involved growing bacteria in a medium containing a heavy isotope of nitrogen, 15N, and transferring them to a medium containing a light isotope of nitrogen, 14N. The bacteria were allowed to replicate, and the DNA was extracted and centrifuged in a density gradient. The results showed that after one generation, the DNA was a hybrid of 15N and 14N, indicating that the DNA had been replicated semi-conservatively. After 2 generations, the DNA was a mixture of 15N, 15N, 15 and 14N, a mixture of both, and another one, heterozygous 14N, 14N, confirming the semi-conservative model of DNA replication. The Michelson-Stahl experiment provided strong evidence for the semi-conservative model, which states that the original DNA molecule is used as a template for the synthesis of a new DNA molecule, resulting in 2 DNA molecules, with each each with one old strand and one new strand. So obviously that was, I mentioned it earlier with Avery, right? How one strand was able to take on another strand of virulent bacteria. So it's basically the same principle. So you have the virulent strain, right? A double helix, it unwinds, and then you get the good strain. It has a double helix, it unwinds. So you have four strands, and these four strands each make a new strand, right? And you get basically a semi-conservative model of both, eventually forming a virulent strain of bacteria with non-virulent strain of bacteria DNA, basically a 14-15N. So the gene-to-gene protein dogma, also known as the central dogma, is a fundamental concept in molecular biology that describes the flow of genetic information from DNA to proteins. The dogma states that DNA is transcribed into RNA, RNA is translated into proteins, and the gene-to-gene protein dogma was first proposed by Francis Crick in 1958 and since become a cornerstone of molecular biology. However, it is not always correct. Limitations to the gene-to-protein dogma. While it provides a general framework for understanding the flow of genetic information, it has several limitations. Epigenetic modifications such as DNA methylation, histone modification can affect gene expression without altering the underlying DNA sequence. Alternative splicing allows for the production of multiple proteins from a single gene, challenging the idea of a one-to-one correspondence between genes and proteins. Non-coding RNAs, such as microRNAs and siRNAs, play important roles in regulating gene expression and can act as intermediates between DNA and proteins. Environmental influences, environmental factors such as diet and stress can affect gene expression, protein production, highlighting the importance of considering the interplay between genetic and environmental factors. I believe that's something that we're going to be talking about quite heavily throughout this semester. While it does provide a useful framework for understanding the flow of genetic information, it is not always correct. The complexity of gene regulation, the influence of environmental factors highlight the need for a more nuanced understanding of the relationship between genes and proteins. There's some exceptions to the dogma of reverse transcription. Some viruses can convert RNA back into DNA. Epigenetic regulation, as we stated before, environmental factors, they affect gene expression without affecting the underlying DNA sequence. So just phenotypic expressions without actually having to modify the gene. So DNA in the universe. The stability of life refers to the ability of living organisms to maintain their genetic information and pass it on to their offspring, right? That's Darwinism 101. And it's ensured by DNA replication. And DNA replication has an error rate less than 1 and 10 to the power of 9, a very huge number, or very small number. DNA repair cells have mechanisms to repair damaged DNA, such as base excision repair and nucleotide excision repair. Genetic recombination during meiosis helps to maintain genetic diversity while ensuring the stability of the genome. The mutability of life refers to the ability of living organisms to change their genetic information over time, allowing them to adapt to changing environments and evolve into new species. This mutability is ensured by mutations. Errors during DNA replication or repair can lead to mutations which can result in changes to the genetic code. Genetic recombination during meiosis can lead to the shuffling of genetic material, increasing genetic diversity. The movement of individuals with different genetic makeup into a population can lead to the introduction of new genetic variation. The double helix structure of DNA provides stability to the molecule. The pairing of nucleotide bases A, T, and C, or A to T, G to C, provides stability to the double helix. The hydrogen bonds that hold the double helix together, the sugar phosphate backbone, provides a stable framework for the double helix. These were all mutations that occurred at one point or were set from the get-go. So its structure and mutability allows for base pairing errors. This can lead to mutations. Uh, Base modifications, chemical modifications to the nucleotide bases can affect gene expression and lead to mutations. DNA recombination, when your parents made you. I like to think that I'm like some superhuman that holds the key to like a major disease that's going to happen in the future, and I'm going to be the only person in the world that's immune. I don't know, maybe it's just a book that I should write. But in conclusion, the stability and mutability of life are two fundamental aspects of biology that are intimately and 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 so tightly linked to the structure and function of DNA. The double helix structure of DNA provides stability to the molecule while also allowing for mutability through its base pairing errors, base modification, and DNA recombination. So yeah, I think mutations are possibly our greatest asset as species. Any species in particular can always mutate. COVID-19, for example, mutated and became a stronger virus. Eventually, our immune systems recognized this mutation and became even stronger. So it's an ongoing library of mutations. Um, and I think this is, in fact, going to be the key to medicine. I saw a show that was particularly interesting, Alice in Borderland. Um, they put these people kind of like in a matrix-like dream state, um, and they showed them this reality where disease no longer existed, where humans were like just sentient beings, almost floating around the universe, um, and basically gene therapy just became the norm for everybody. It became normal for everybody. There weren't any more sicknesses, no more diseases, no more anything that could affect a mental stability. Everything was deleted or made stronger through gene therapy, right? Which is genetics future, the future of genetics, in my opinion. But mutations, I think, hold the key to everything that could go right or could go wrong for humanity. Sorry about that. I have. Ugh, ugh. Um, so, in conclusion, the double helix provides stability, it's efficient storage, easy replication. Um, it has major and minor grooves. It's base pair stacking, physical structure and function. It's genetic information storage, replication, transcription, translation. Um, DNA is an extremely stable molecule, extremely stable, and it looks super cool. If you ever worked with a banana in electrophoresis to ever extract DNA from a banana, it is a really cool structure to see live and to work with it is it's an incredible experience. If you've never seen it, how it looks like in actual real life, not just a diagram where they're showing it to you, I suggest you look at videos like that and really puts things into perspective and puts things into perspective how all these people that came before us actually discovered these things and worked with these things and put the time and effort in order to make the discoveries that we know today and eventually advance medicine. I think that's a major branch that a lot of us are trying to reach, or most of us. Maybe you're not heading in that direction. Maybe you just want to work in research, which is something that I'm fascinated by as well. Um, so I hope you like the format of these videos. I think I'm gonna change a little bit how I started this particular one and just the direction that I want them to take. But I think this is how they're gonna be going from this point forward. So Thank you guys for stopping by. I hope you guys enjoyed, and I hope you learned something from me. See you guys in the next one.